Hello, everyone. It is a pleasure to be part of this great conference. So I'd like to thank the organizers for the opportunity to share my experience on the future of clinical relevant dissolution testing and physiologically based biopharmaceutics modeling, PBBM, PBPK, in drug product development, manufacturing changes, and controls. What I like to do today in this presentation is to tell you a short story on how the solution testing regulatory application is evolving, starting with its current application as per guidance and moving into the current trends on the application of the solution testing. I will talk about what data are needed to establish clinical relevant dissolution testing and how can clinical relevant dissolution testing information be leveraged in support of drug product development and regulatory decision making. I'm going to describe the role of PBBM and PBPK in drug product development, which can be summarized as building a safe space. And I will list some general expectations on regulatory submissions containing this kind of data. I will switch gears and describe the future role of clinical relevant dissolution testing and PBBM, which I view as a stepping stone towards patient-centric drug product development, enhanced drug product control strategy, and regulatory flexibility. I will touch upon some challenges in the implementation of these tools, which might resonate in your mind, and hopefully, well, it will serve as an opportunity to spark some ideas in terms of what is needed to fill in the gaps in knowledge in these fields. And I will conclude my presentation with some take-home messages and future directions. I'd like to start this presentation by reminding you about the FDA vision in terms of advancing drug product quality. And there have been several FDA initiatives, one of which is the pharmaceutical manufacturer in the 21st century. And according to Dr. Janet Woodcock, it refers to an efficient, agile, and flexible pharmaceutical manufacturing sector that reliably produces high-quality drugs without extensive regulatory oversight. Another initiative is patient-centric drug product development, which is part of the 21st Century Cures and PDUFA 6. About 2 or 3 years ago, the Office of Product Quality published the One Quality Voice brochure that describes the establishment of acceptance criteria for CQAs based on clinical relevance instead of process capability or manufacturing controls. So the question is, what is the role of dissolution and PBBM, PBPK in this effort? And just to put into perspective how regulatory agencies like the FDA value or see the role of modeling and simulation and regulatory decision making, let me share with you what the ex-commissioner from the FDA, Dr. Gottlieb, presented back in 2017. He said that we are on a sustainable path where the cost of drug development is growing enormously, and that we need to do something about it now to make the process less costly and more efficient. And one way to solve this is the inclusion of more modeling and simulation in regulatory decision making. So before I start talking about the future of dissolution testing and PBBM in support of CMC applications, let me talk about what has been the past and the present use of dissolution and some current trends in its application in regulatory decision making. I've listed here several FDA guidances and some federal regulations that mention the application of dissolution testing, starting with the Code of Federal Regulations, obviously not a guidance, which mentions dissolution testing as one of the drug product specifications necessary and required to ensure quality. We are familiar with the SUPAC guidances, which list dissolution as a tool in support of minor or medium risk manufacturing changes. Also, the BCS and IBIC guidances, which list dissolution as a surrogate for bioequivalence and bioavailability studies via waiver request. And for this to happen, of course, certain data are required beyond dissolution. A couple of these guidances, such as the SUPAC and IBIC guidances, were published more than 20 years ago, where the application of QBD principles were not integrated per se in the pharmaceutical industry. And so the application of dissolution testing reflected the needs and tools available to the pharmaceutical industry at that time. So, however, with the advent of quality by design, there has been a trend in several regulatory applications of dissolution beyond those listed in the guidance I described earlier. And what I've done here is to somehow dissect the concept of QBD, and I tabulated its parts to identify in more detail several areas of dissolution application. And as you may know, according to the ICA guidances, especially the Q9 and the QBR2, QBD starts with the predefined objectives, which I see as defining the quality target profile and identifying critical quality attributes. And as you may know, the solution is usually identified as a CQA, especially for modified release products. QBD emphasizes product and process understanding, and this can be translated into the identification of CMAs and CPPs of the final product that must be controlled to meet the target quality profile. And in this case, you may know that the solution can be used to identify the CMAs and the CPPs. It also emphasizes process controls, which means the development of appropriate control strategy, including justifications. In this case, the solution can be used to verify the design space, the limits of the design space. And this control strategy can be guided by risk assessment dissolution. And dissolution can support this effort by the establishment of clinical relevant drug product specifications and a risk mitigation strategy. Well, despite of the all well-known value of dissolution testing in drug product development, I would say that there is a dilemma, and this dilemma is that its recognition as a true and key enabler of enhanced drug product understanding is precluded in many cases by the uncertainty of its predictive ability, especially for modified release formulations. So the question is, how can we remove this uncertainty and what data is needed? Right, I see this data needed as part of two parts of a puzzle. And the first part is the establishment of a relationship between the CMAs and CPPs variations and dissolution. And the other is the establishment of a relationship between dissolution and in vivo impact. You might be thinking, well, why is this important to establish this in vitro in vivo link? Well, the thing is that without an understanding of the relationship between these critical attributes, process parameters, dissolution, and clinical outcome, the method could be under and or over discriminated. And if the method is under discriminating, it might allow the release of drug that is out of specifications, posing a risk to the patient. On the other hand, if the method is over discriminating, the risk for rejecting there is a risk for rejecting good batches, good product batches. So I'm going to focus on describing the data needed to establish the relationship between dissolution and clinical impact. And to establish this relationship, what is basically needed is information on different dissolution profiles from manufacturing variables that are tested in clinical PK studies. And this in vitro in vivo information can also be used to develop a conventional IVIVC or physiological based IVIVC or IVIVR. And the beauty of these approaches is that they empower dissolution testing to know its boundaries, out of which batches will be rejected, leading to biopredictive or clinical relevant dissolution testing. So what I'm going to do next is to describe what is the role of physiologically based modeling in support of drug product quality specification setting, and what are the regulatory expectations by the scientific community. And a discriminated method may or may not be biorelevant and vice versa. All depends on whether biorelevant media or biorelevant hydrodynamic conditions are considered, and the method is discriminating. These two types of dissolution methods can be considered as regulatory methods, but may or may not be clinical relevant. But if there is data, if there is no data available linking product quality variations to dissolution and in vivo performance, but specifications are set closely based on the performance of the bio batch or the pivotal clinical batch, then the clinical relevance may be established. But in this case, it's not always assured, especially for modified release or BCS class drugs, BCS class of 2 or 4 drugs. Of course, that will depend on whether the method is under and over discriminated. When there is data available linking drug product quality, dissolution, and in vivo performance, for example, via BE studies, then they can become clinical relevant. And when these two methods are developed considering model and simulation, such as IVIVC or PBPK, then they can be biopredictive. And these methods are considered or classified automatically as clinical relevant. I encourage you to take a look at this reference, which give more detail on the terminologies for dissolution testing. So now what I'm going to talk about is the role of PBBM and PBPK in support of drug product quality. And the beauty of these physiological-based modeling strategies is that they are predicated on leveraging the available knowledge and experience by pulling all the data that are available, such as solubility data, metabolism, preclinical and clinical data, and especially input for formulation variants, which is usually done via dissolution testing. And then when all this is done, then it allows for the possibility to create robust predictions in vivo performance of drug products. So, and then one important feature of physiological-based modeling, PBBM, and that differentiates it from purely PBPK is the identification of the establishment of safe space, leading to the reduction in the number of studies needed and the establishment of clinical relevant drug product specifications. So what is safe space? I have mentioned safe space in several of my slides, and I will do so in the slides that I'm going to present after this. And safe space are considered as the boundaries defined by in vitro specification, in this case dissolution and other relevant drug product quality attributes, within which drug product variants are anticipated to be bioequivalent to one another. And just to explain this concept a little bit more, let's assume that we have product variants with distinct dissolution profiles, so that when tested in vivo, the blue and the green were found to be bioequivalent to each other, but not to the black profile. A safe space can then be defined on the extremes of dissolution profiles that were found to be BE in such a way that any major or further changes can be justified without additional bioequivalent studies. And if you pay close attention, I said any major changes implemented to the drug product. The question is this always the case? And I would say that this is the case as long as there is a thorough understanding in the relationship between the CMA CPPs and dissolution and their potential interaction, and the dissolution data generated from these, let's say, DOE studies were used to establish this safe space. Now, what are the applications, common applications of PBBM in support of drug product quality? And I've summarized some of them here. And these are some applications that I saw while I was a reviewer at the FDA. And it can be used, PBBM can be used in the establishment of wider acceptance criteria that are clinically relevant and to demonstrate the biopredictive ability of the method. It can also be used to establish clinical relevance specifications for CMS and CPPs, such as particle size distribution, polymorphic form, and hardness. It can also be used to justify bio-waiver requests and other formulation effects, such as the effect of food and formulation changes, such as in PPI interactions. So, how common is the use of PBVM? I presented this slide last year while I was at the FDA, and this slide shows the experience in PBBM in support of drug product quality. And based on a database created gathering data from 2008 and 2018, the agency received about 30 submissions containing PBBM, and the majority of these were in support of drug product specifications such as dissolution. I want to emphasize that the majority of these submissions were received from 2015 and 2018, which indicates how agencies such as FDA are embracing model and simulation tools in support of drug product quality. Even more so, Dr. Sally Cho, the director of OGD, mentioned on her presentation in the 2020 public workshop on generic regulatory initiatives that the use of PBPK modeling to support approval, the agency used PBPK modeling to support approval of locally acting drug products. Now, what I'm going to do now is talk about what are the general expectations on submissions containing PBBM. And it's important that the report includes a summary that informs the assessor of what is the bottom line and clearly defines what is the purpose of this analysis, what is the problem, what data are needed. It should also include the modeling and simulation objectives. In other words, what is the regulatory question that you are answering that led to the conduct of this modeling exercise or analysis? It should clearly describe in detail the model development that includes, for example, the model structure, the disposition model, and a mechanistic framework of absorption modeling and what data was used. For example, a combination of a mechanistic absorption model with a simplified compartmental dissolution model might be acceptable with justification. And since models are being leveraged to justify CMC changes, it's really critical that it describes the approaches taken to integrate dissolution data. Why? Why is that? Well, because dissolution, as I mentioned before, is the only in vitro parameter that provides insight to the rate and extent of in vivo performance of the changes implemented. Also, this report should include an explanation for modeling assumptions. In other words, assumptions underlying model structure and parameters should be clearly presented, specifically in terms of the disintegration and precipitation, potential for precipitation, degradation, transport and first pass effect, distribution and clearance. And finally, the report should also include a rational and supportive information on model parameters. Well, when it comes to model verification and validation, the predicted performance of a model should be validated for its intended purpose. What do I mean by intended purpose? Well, if the model is going to be used to justify CMC changes, then it should be validated with data from studies that evaluated the effect of formulation changes. Data to support the validation should include, for example, graphical and numerical comparisons of the predicted and observed drug concentrations versus time, as well as PK parameter estimates and statistical analysis. All this should be provided as part of validation. Independent data sets not using model development should be included, preferably the use of non-BE data should be included to challenge the predictive performance of the model. Model validation criteria should be established a priori, and in the case of mechanistic IVIVC, it should comply with the criteria provided in the IVIVC guidance. Now, for the sake of saving time, I have provided additional data as part of backup slides, which this data is more specific in terms of PBBM expectations. Please take a look at this at your leisure, since I think the slides are going to be shared. Also, I encourage you to take a look at this article recently published that contains a lot of information in terms of PBBM, its application, validation steps in support of drug product quality. And this article was recently published in 220 by a group of people from industry and scientists from the FDA. I would say that the application of PBBM and PBPK into drug product development is still evolving. It is well known that it is a promising tool, but it is underutilized for risk assessment and establishment of a safe space. In addition, as you may know, there is no regulatory guidance available on the application of PBBM and PBPK in support of drug product quality. Then it's not surprising that the criteria that the scientific community, including regulatory agencies, are still refining the practice and policy on PBBM. And so this is reflected in terms of the common mistakes seen in regulatory submissions on PBBM, and I'm listing some here. When it comes to model structure information, it is usually insufficient. For example, the submissions do not have enough information on mechanistic framework accounting for impact of quality attributes and absorption. There is no justification for input parameter values selected for the drug substance, the PK and formulation variance, and sometimes the physiology either. We have the agencies have seen insufficient data, and in many cases, the program project files are not included. Also, parameter sensitivity analysis is not always provided. When it comes to validation data, in many cases, it is insufficient. For example, usually there is no sufficient data, or the data is inappropriate for model validation. And this is tied to the concept I mentioned before, that the model should be validated in terms of the intended use. Another issue is that the reliability on the simulations results is questionable, especially in terms of virtual BE analysis, where there is uncertainty about the subject variability assessment. Now, based on all the information I have presented today, hopefully you have an idea on what is the future of clinical relevant dissolution testing and PBBM in support of drug product quality. And what I'm going to do now is to summarize this on the next 3 slides. First of all, I see PBBM as a key player in enabling patient-centric drug product development. And just to make sure we are on the same page, I'm going to define what is patient-centered drug product development. And from that perspective is the development of science and risk-based drug products based on the implementation of drug product specifications in process controls and control studies that are clinical relevant. And this will be possible as long as clinical relevant biopredictive dissolution testing, which has been empowered by PBBM or has been used in combination with PBBM, is implemented in drug product development, for example, as part of the DOE studies. The second key application of PBBM looking into the future is as an enabler of enhanced control strategy. And what I mean by this is that the implementation of PBBM early in drug product development allows for building clinical relevance into the whole process, which is critical for programs that follow continuous manufacturing that rely heavily on multivariate analysis and real-time release testing models, including dissolution models for RTRT. And why is this critical for a path follow as I depicted here in yellow? Well, it is critical because relying on a control study that does not include clinical relevant dissolution testing as an endpoint poses a risk to the patient since, as I mentioned before, dissolution is the only in vitro test that proves the rate and extent of systemic exposure. All the applications of PBBM are relevant from a patient perspective, all the ones that I just mentioned earlier. But the question is, what is the benefit for the pharmaceutical industry? What is the incentive for embarking in developing PBBM models? And the answer to this question is one is the possibility for gaining regulatory flexibility via safe space. In other words, the possibility for reducing the number of required BE studies following major manufacturing changes, CMC changes. And to further explain this, let me just spend a few minutes and go over this plot that has several possibilities into building a safe space, somehow explained before earlier. Let's assume that you conducted a bioequivalence study with 3 formulation variants and found that the red profile, which is the target, was bioequivalent to the blue profile, but was bioequivalent to the blue profile, but not bioequivalent to the green profile. And as I explained before, a safe space can then can be created between these two profiles as depicted here. Now let's now assume that the PBBM model was developed, which allow for you to define the limiting dissolution profile that is bioequivalent to the target, such a way that a safer space, a bigger safer space than the original can be then established via this approach. Even more so, let's assume that you originally have data from 4 formulation variants and that you were able to develop an IVIVC which met the internal and external validation requirements. Then we can then build a safe space that gives flexibility in the types of changes allowed a wave, as described in the guidance, such as changes in the amount of excipients controlling the release rate of the product. So hopefully it is clear that the implementation on PBBM allows for the building a safe space which is likely to be wider compared to, let's say, that defined by dedicated PK studies. That I like to end my talk by leaving you with these take-home messages. There are no regulatory hurdles for the use of innovative model and simulation exercises. And in fact, the regulatory agencies, especially the FDA, embraces and encourages the use of model and simulation in support of drug product quality. The development of conventional mechanistic IVIVCs is always considered the gold standard for gaining regulatory flexibility throughout the drug product life cycle. However, if this path ever fail, let's say you are unable to develop an IVIVC, this data generated during drug product development, along with any other PK studies that you conducted, may be leveraged to define a safe space via, as I mentioned before, IVIVC or mechanistic IVIVC, IVIVR via virtual bioequivalence studies. And finally, building a safe space is not only relevant for gaining regulatory flexibility, but a stepping stone towards setting clinical relevant drug product specifications and patient-centric drug product development. And with that, I'd like to conclude my presentation, and I will be happy to entertain any questions that you might have. Thank you again for your attention.